So hello, everybody. On behalf of the Simon Center for the Social Brain, I want to welcome you to our webinar-based lunch talks series. I'm Reganka Sir. I'm the director of the Simon Center at MIT. And we have been pleased to be able to continue with some modicum of normalcy our community events, which include the lunch series offered by Simon's postdoctoral fellows, as well as researchers who are active in the Simon Center targeted projects, as well as our colloquium series, which has taken place on Wednesdays at 4 p.m. So we have had 2 colloquium series talks, and this is the second of our lunch series talks since we went into a social distancing mode. First and foremost, I hope every one of you, and I see there are 64 attendees for this webinar from not only Boston, but all across the United States, and I recognize some names from abroad as well. So welcome to all of you. I hope you are staying safe and healthy. And before I give over to Goping Feng, who will introduce Will Menegas, who's our speaker today, let me go to the second slide and go over a few housekeeping items. Will, can we go to the second slide? All right, so Goping will be our moderator today, and we have disabled the chat and the raise hands function because we find that questions are best done through the Q&A button. So click the Q&A, add your question to the field. If more than one person likes the question, upvote it, and then Will take a break or two during his presentation because we find that works well in order to break up you staring at your screen and Will staring at his screen into shorter segments. And of course, at the end as well, he will be happy to take questions. So with that, I'm going to request Goping to introduce Will Menegas.
Hi, everyone. Welcome to the Simon Center Lunchtime Seminar Series. I'm Goping Feng. I'm a professor of neuroscience at the McGovern Institute and in brain cognitive science. Will, I'll give you a brief introduction of Will, today's speaker. He's a postdoc fellow in my lab, and then he will take over to give the seminar. Will attended the University of Chicago for his undergraduate study, majoring in molecular and cell biology. Then after graduating from University of Chicago, he joined Noshige Uchida's lab at Harvard as a PhD student, and he did some phenomenal work in his for his PhD thesis, studied the special function of dopaminergic signaling in the posterior striatum, use a mouse as a model, and combined a variety of in vitro and especially in vivo technologies. He published his thesis work in 2 eLife papers and 1 Nature Neuroscience paper. So after his PhD, he approached me. He wanted to study, use marmoset to study neurobiological mechanisms of social behavior and the social dysfunction in ASD. So that was that time was still very new for us, even in the lab with the marmoset. So I thought, oh, this person is really brave. And you all understand that non-human primate work is much slower, longer. And he being in the lab almost 2 years now, you will see he made a tremendous progress. And he probably at the beginning, he was shocked because he come to the lab, he want to study social behavior, using in vivo recordings and the behavior analysis. And he probably realized, oh man, you don't have anything. We literally even didn't have a program to automatically tracking the behavior to quantify the social behavior. So he actually single-handedly started this program, made a really important progress. Now he has the program he will talk about today. And he also made a progress in how to use the in vivo recording to study social behavior in the future. So without further ado, I will pass to Will. Will, it's all yours.
Thanks for that really nice introduction. Like Goping said, I've been here about 2 years in the lab as a postdoc. And so today I'm going to tell you guys a little bit about what I've been working on in that time. And so first, a little background about the lab and Goping's approach. Goping is taking a really heroic task on, which is to create genetically modified model organism marmosets, the way that people have been doing a long time with mice. And the basic strategy here is to first create these animals using gene editing, then see how they are different from normal animals, apply a drug or therapy, and then see if that reverted their behavior in some way. And so a crucial part of this pipeline is to be able to quantify behavior, including social behaviors, both in this validation step for the model organisms and in the step to see if a treatment is effective. And if this pipeline looks familiar, it's because this is essentially quite similar to the work that they've done already, or that the Feng Lab already has done in mice. In this case, knocking out SHANK3, which is a gene expressed in the striatum, observing this difference in behavior where the mice are no longer interested in a novel animal, restoring the protein, and then observing that the behavior is reverted, so back to being interested in a stranger. I think this approach was so successful because people have such a good understanding of, or at least we think we have a pretty good understanding of mouse behavior and how to probe it. But if you're starting from scratch in a new animal, it's maybe less clear where to start, and you might need to take more sort of untargeted, unbiased approaches. So I looked to people using these sorts of approaches in mice studying, for example, SHANK3. In this study, the group looked at all of these kind of human-defined behaviors and quantified the frequency that mice did these behaviors, where here one is baseline, and so here is below baseline and here is above baseline. And they found that mice did more of this stretch attenuate posture behavior, this kind of cautious exploratory behavior. And so they wondered if perhaps their fear or caution around novel animals was related to kind of a general fear or cautious phenotype. And they did a targeted assay where they just put one of these, if you work with mice, very familiar objects, but to these mice, novel objects in their cage, and found that normal mice kind of explore it, aren't interested in it, whereas SHANK3, mice lacking SHANK3 were very afraid of that object, kind of retreated away from it. So I think it's a really cool instance where kind of a unsupervised, or maybe not unsupervised in this case, but a more unbiased approach gave you the exact same kind of conclusion as a very targeted approach. And so I got interested in, okay, how can we study behavior without having very targeted assays? And so I kind of just looked at the field. Here's just a few examples of people who study in other model organisms like worms, flies, zebrafish, and mice. I wondered just kind of, what are these guys doing? And basically, a lot of these researchers and labs have converged to a similar approach, which is to first represent the animals in a really low dimensional space. So for example, I guess the worm is pretty well represented by like a few sine waves, flies by principal components of the image. This group represented the zebrafish by like key points on it, like the eyes and some points on the tail. The data lab represented mice by PCA on the raw image data. So there's slight differences in the exact implementation, but the basic idea is to take the video of the animal and reduce it from these thousands and thousands of dimensions of the image into a few manageable dimensions. So we can study how those few dimensions move over time, because the time dimension will ultimately kind of explode the data size. And so these groups all take different sort of either model fitting or dimensionality reduction to look for patterns in how these low dimensional representations of the animals move over time, with the idea that sort of repeated motifs in these will correspond to human recognizable behaviors. So for the worm, it could be something like turning clockwise or turning counterclockwise. For the mice, it can be something like rearing or darting, things that a human should be able to name that recur over and over again. So my goal for the talk is to apply, or my goal for at least the first part of the talk is to apply these methods to the marmosets. Does anyone have questions so far? I don't see any in the window, so I'm going to keep going. So the basic setup here is that we installed glass ceilings and cameras over a lot of the cages, looking downward into the cage. And then we colored one of the animals in each pair's hair so that we could distinguish the two animals from each other. And I just collected a lot of data in this format, this kind of like 1,000 by 1,000 pixel, 30 frames per second data stream plus depth. We recorded many animals in many facilities. In total, at the starting point for kind of looking for patterns, I had over 100 million frames and over 15,000 hours of video. So the first thing for this was to train a network, like a deep lab cut network, to recognize the two animals in the image. So here you have some key points on the animal with blue hair tufts and some key points on the animal with white hair tufts. After labeling a few thousand frames, the network reached asymptotic performance on labeling. And after doing a little bit of filtering, the network reached quite good accuracy on all of the data points. And so here's kind of a trace of how each of the animals moved over like a 15-minute period. You see this animal, the red trace kind of ran around a lot. The blue trace was less active and kind of hung out in a few spaces. Here's an example of labeled data. Here the closed circles are the animal with white hair and the open circles are the animal with blue hair. These traces are like the last 1 second, I think. And so you can see even as they kind of move around the cage, the tracking is pretty good. At least good enough to start trying to look for patterns with some of the approaches that I talked about before. And so the kind of important key points here are the blue points, the head, the black points, the body, the shoulders and hips, which are red and green. And when the limbs come out, you can see them in cyan and magenta. Okay, so if you just look at this data set in kind of a dumb way, just look for in the traces, the principal components of variance, what you find probably, as you would expect, are that two components really dominate the, or really explain most of the variance, like 95%. Those two components correspond to the animal's X position and the animal's Y position. Then two more components explain the remaining like 5% of the variance among the frames. Those components are corresponding roughly to the body angle. So like sort of all the other studies, I thought, okay, we'll just subtract out the X position, Y position, and body angle, and rotate and center the animal in this data stream so that the animal for every frame roughly looks like this. It can like turn its head and extend its arms and do things like that. But the X, Y position and rotation of the body are subtracted. And also to correct for differences when the animal is nearer or further from the camera, we also normalized to the head radius, which should be a fixed distance for each animal. Here you can see that that head radius takes on many different pixel values. And if you normalize by it, you get less dispersion in distances between body parts, which makes sense because they should be roughly a fixed distance from each other. And this agrees well with the depth information, by the way, this head radius measurement. Okay, so once kind of all these things have been subtracted out, now the principal components of the data set are actually kind of like human recognizable postures. So like here, principal component 1, as you go down it, you see the animal's head turns. Principal component 2, the body extends. Principal component 3 is like a climbing posture. Principal component 4 is like a marking posture. And importantly, these postures or these principal components vary on really short time scales. Like they take on a lot of values over 1 second time scales, but they on average are 0 over the course of an hour. So that means we're not getting components like, for example, if one animal is larger than another animal, you might expect that a principal component could be the body length or something, but we're not seeing things like that. Instead, we're seeing these quick timescale changes in posture, which is good. And just a few notes on kind of expanding this data from the initial kind of like deep lab cut style points. One thing is we used wavelet representations of each body part. The logic here is that if you have some signal, like this signal, you can use a wavelet transformation to visualize structure on multiple timescales for each, like each observation, I guess, in the array. So like here you have information about the moment. Here you have information kind of on an intermediate timescale, here on a long timescale. And that's important because behaviors could evolve over multiple timescales. So we want to have all that information when we do any of these analyses. And secondly, having the second animal this is a big complication. It's the subject of a lot of study. But just a brief summary, I used a lot of terms that would relate the two animals to each other, such as the distance between their bodies and heads, the difference between their body angles and head angles, their head angle relative to another animal. And essentially a lot of this is basically copied from people who study social behavior in flies. And so kind of what I ended up with here is these feature matrices. Here each row is a frame and each column is a feature. You can already see, just looking at it, there's some kind of structure to it. And I just used TSNE to look for the kind of largest scale structure and found 3 like large clusters in it. Are there questions up to this point? I don't see anything in the question answer, so I'll just keep going. So I got these 3 kind of large clusters. Here you can see how they are organized over time in an example recording of about 5 or 6 hours. You can see that cluster 2 kind of is in these long chunks, and cluster 1 and 3 are interspersed in the time between those large chunks. When I looked at histograms of the animal's velocity and the distance between animals, I could see very clearly that cluster 2 corresponds to when the animals are near each other, and cluster 3 corresponds to when the animals are moving really quickly. So what you can see about this kind of just visually is that the animals sometimes sit next to each other and don't move for long periods of time. And then other times they're in this kind of more active mode where they're more on their own and they're more moving around. Animals kind of cycle between these two modes, this active moving around, not near each other mode, and this sitting and next to each other and not moving mode. If you look at the distribution of these three T-SNE clusters within the image, you can see that they kind of spend time alone all over the cage, and they run between these places. And there's a few places that they sit next to each other here in magenta. There's also some places where they never sit next to each other. For instance, they never sit next to each other on this log. They never sit next to each other in this kind of circular space where they can view outside. They sometimes are climbing next to each other. Sometimes they're alone in this area. So you can see there's kind of rich structure in terms of the spatial information as well. When I measure the usage of these over many recordings of many animals, you can see that there's some clear patterns. So here every tick mark is 1 day of recording. It's sampled evenly, I think like 300 days from 100 animals. You can see that, oh, actually these labels are switched, that's a mistake. This is the fraction of events. So most of the events are these alone events and these moving events, and a smaller number are these sitting near each other events. This is the fraction of time that the animals spend in each of the types of event. So most animals spend most of their time just sitting by themselves, a decent amount of time near the other animal, and then a very small amount of time in the in this third cluster where they're running quickly, which makes sense because they can't just run around all day. So what's really notable is that although running is a large fraction of events, it's a really small fraction of time. And these kind of trends are consistent across all our recordings and all our animals. So now what we can start to do is design tasks or look for differences between animals based on their genotype or some other manipulation, where we have like some controls, some manipulation, and then some time that we're measuring to see a difference. So in this case, I recorded their behavior for 2 hours. I briefly separated the animals, and then I reunited them and recorded their behavior for another 2 hours, wondering whether they might be more likely to kind of like stay near each other if they were briefly socially isolated, as we've seen in other organisms over and over again. And we did see this. That was the case. There was a change in the fraction of time spent such that they spend less time alone and more time near each other. It didn't change the amount of time they spent running, and it didn't change the kind of fraction of each type of event. So the interpretation here is that the animals spent less time per event standing around alone and more time per event sitting next to each other. So we have some sort of basic readout of how this task influenced their behavior. But it's kind of crude, and so we want to look a little bit more in depth, and in particular, we want to be able to characterize these chunks into more than just 3 clusters. So that's what I'll talk about next. I do see there's a question, so I will answer it. Daniel Gibson asks, how do you know whether to use PCA or T-SNE? That's a good question. They're both dimensionality reduction. I had basically used PCA to look for patterns in the already low-dimensional posture data. I used TSNE on the data set with lots and lots of dimensions, because some other people, like Gordon Berman, for instance, had found kind of a lot of really nice clusters using that method. I only found 3 really nice ones, and then there's some subclusters, but I didn't think they were that clear. Another question, since the behavior of the monkey seems to be affected by environment, do you envision that future behavior experiments have to be done in a standard cage with the same setup, or have the same elements like log, window, etc., where the data sets are not comparable? That's a really good question. Certainly, most of our cages have that similar setup. And yeah, actually, it's even worse than what you're imagining, because even different rooms have different kind of neighbors and different number of animals, different like visibility of other animals. So yeah, those environmental factors are huge. I guess the best thing is for kind of controlling the experiment by comparing like a brief window before the perturbation and after the perturbation. Or in the case of modified animals, perhaps controlling the experiment by having wild type animals and genetically modified animals housed in like extremely similar settings, maybe even in the same room, and then comparing these two directly. Because as you're saying, all these features of the cage are gonna impact kind of the distribution of their behaviors. Oh, and also people coming in and out certainly will influence it. So for instance, imagine the animals are in one of these bouts where they sit next to each other for a long time, but then people start coming in and out of the room a lot. Certainly that'll affect their probability of exiting that relatively like calm behavior. For my part, I just tried to record a lot and then hoped that things like that would average out. Okay. So I wanted to look for a few patterns in the relationship between the two animals. And so one obvious thing to look for is their body angle relative to the other animal. And what I found is that when the animals are alone, so the other animal is in one of these positions here in black, there's not much pattern to their body angle relative to the other. Conversely, when the animals are really near each other, there's a strong tendency for them to be sitting side by side with their bodies aligned. So now I separate these histograms into bodies aligned, bodies unaligned. You can see that when the other animal is in front of them, their bodies tend to be unaligned. So if the other animal is in front, they tend to be kind of oriented towards the other animal. Whereas when they're side by side, their bodies tend to be aligned. So for instance, side by side, sitting next to each other like this. So again, if we look across our like 300 days, we see there's no trend when the animals are alone. We see there's a very obvious trend when the animals are near each other. This trend is present in every single day except for maybe 3. The extent of it varies a little bit, probably along kind of normally distributed along here. So what you see is that here when the animal's in front, they tend to be facing it, and when the animal's on the side, they tend to be sitting side by side. When the animal is behind them, there's again no trend. The other thing I wanted to look at is the head angle relative to the other animal. Marmosets luckily have these really easy to spot ear tufts that makes it easy to kind of guess what direction they're looking in. So maybe this animal could be looking at the other animal, whereas this one probably is not. What I found is that whereas the other animal's position is randomly distributed when the animals are far apart, the head angle relative to the other animal is not randomly distributed. So there's a strong tendency to look towards the other animal. And that's present again in like all of these sessions. So it's a very strong trend. And what I see is that regardless of where the other animal is, here in like these degree-based quadrants, you see that the animal looks towards it. So within this kind of 30-degree range of the other animal, way more than you would expect based on fitting the line, based on this other part of the line. And so even when the animal is almost directly behind them, they look at it way more than you would expect them to. And so basically all of these add up to give you this histogram that you see here, or this rose plot rather. If you look really carefully at the head movements, you can see they tend to look in a direction and make sharp bouts to change their head bearing. So it's really clear when their head is still versus when it's rotating if you look at the head velocity. And these types of bouts, I just sorted them based on whether they're looking at the other animal or not before and after the bout. So you can see here this type where they kind of look away and look back, where they're not looking at the other animal, or where they start out looking and then look away, where they start out looking away, end up looking towards, and when they look in two random directions, neither of which are towards the other animal, there's a strong tendency that most animals make most of their head movements in this type 4, which is an undirected head movement, whereas we already saw that almost all animals spend way more time looking at the other animal. So kind of like in the bulk movement data, we see that the animal's more likely to linger on the other animal once it kind of looks at it, rather than continue moving its head around. It's not that it continually kind of tracks the other animal all the time. So I wanted to take a lot of these features, some of these continuous features I've talked about, and some binary features, like whether the animals are near each other or not, and start to use a model to describe behavior in a more finely granulated way than just these 3 clusters. So the strategy I took is to take the large feature vector that I was talking about and try to fit different types of models, like in the example I'll talk about today, a hidden Markov model, and then observe what the states that it identifies are and see if they're human identifiable. So in this case, the model identified 11, or is it 12? 12 states. I false colored them. These are states where the animal's alone, moving, or near the other. These are the transitions between states. I'll skip over this in the interest of time. Basically, when a human observes these states, they can describe them as the animal sitting around or climbing, turning its head or marking, or in some of these social states, like sitting next to each other with the bodies aligned. And now we can start applying this model to different tasks or different genotypes, and trying to see how the usage of states and the transitions of states change based on the task or based on the genotype. And so importantly, I just want to point this out, in all of these cases, the kind of like definition of the state didn't change, but its usage or transitions did change. That's an important feature. So here we have a control case where I videotaped for 2 hours, waited a while, videotaped for 2 more hours, and you see that the usage of all of these states stays the same. The transitions also stay the same. here we look at the difference between male and female. You see that there's two things, two states are more prevalent in, I think, more prevalent in females. And we can see this is okay, state 5 of non-social and state 3 of social. So we'll go to our lookup table, we'll find that females do more marking. And state 3. And also do more of this kind of grooming behavior on the other animal. And I want to focus more today on these two tasks that I designed, a brief social isolation and a task where the animals can receive a liquid reward for participating in a task. So in this case, I record for 2 hours, split them up, reunite them. In this case, I record for 2 hours, but importantly during this time, the animals don't have access to their normal source of water. So it's a little bit different than the usual control, but not too different because their usage rates of that water thing is pretty sporadic anyways. Then I set up the task and then I let them perform it. And you can see that in both of these cases, the usage of the states has changed, and also the transitions between states has changed. And now we can start to look at these changes and these patterns and really understand what's happening differently in the task than normal. So in this case, the case of isolation, I see that there's kind of across the board less of these non-social states and moving state. And there's a change in the usage of these social states. So they do more of state 1 and less of state 5. So let's look that up. We see that means that they do more of this behavior where they sit next to each other with the bodies aligned, and they do less of this behavior where they're near each other but not really interacting with each other in any way. And you can also see a change in the transitions where there's an increase in transitions towards this state 1. So basically, what we can conclude is isolation across the board makes them more likely to transition into this resting near the other animal state. Now in the case of the task, you can see that that resting near each other state has decreased, and instead now they're doing a few other things. Like for example, they're more active during the task. That's this cyan dot. They also do a few more of these non-social states, but it's not sort of across the board. So they don't do more of state 4, they don't do more of state 6. We can look that up. They don't do more kind of head turning, and they don't do more marking and looking at the other animal. And so in general, this kind of setup is what I'm planning to apply for the genetically modified animals to try and understand if there's different patterns in their usage or transition between these very simple states. I see there's some questions, so I'll answer them. Someone asks, have you looked at vocalization patterns as well, especially after brief separation? Someone says this could be used to study a dynamic relationship between behavior, cognition, and communication. That's a good point. We do have the vocalization data, and that's something that Rohir Landman is working on, and he is going to look at the correlations between these sort of overt behavioral states that we can measure by videotaping them and the vocalizations, which are an important output that they use for socializing and communicating. We also have, is head angle direction or gaze following active vision more appropriate? It's actually quite difficult to check whether they're gaze following the other animal, because it's hard to know exactly what the other animal is looking at along that trajectory, like whether they're looking at something near them or far away from them. So I haven't really addressed that very well. We also have, what is the relationship between monkeys? Is this consistent in the data set, male, female, siblings, etc.? Do you observe any difference in behavior depending on relationship? And that's a good point. In my data set, almost all the animals are a male-female pair. And that's just because that's what we have access to. So our facility is really geared towards this transgenic project. And so the most available animals are male-female pairs that are breeding transgenics. Once transgenic infants are produced, we kind of don't want to mess with them as much. So the animals that I really had a lot of access to, I was really lucky to have access to a lot of animals, were these male-female pairs. I do know from just sort of anecdotally recording siblings, infants, old animals, etc., that there are large differences. Like, for example, the infants are extremely active compared to the adults. So I don't think they can really compare to cross. I also have a question. Do you see any marked behavioral state transitions during female estrus cycle? That's a good point. I do see it by eye that once or twice a month, there's a really big increase, like a really large marked increase in the amount of mating. But my data set still amazingly isn't big enough to capture those patterns very well, because I didn't do a lot of longitudinal videotaping. Rather, I sampled a lot of animals for weeks at a time. Okay, so I have about 15 minutes left. So I'll tell you guys briefly about this work I've been doing to record neural activity in these freely moving and socially interacting animals. The setup is to implant these silicon probes on a drive that has 5 millimeters of travel, such that it can go through dorsal and ventral striatum over a long time. The amplifiers are housed permanently on the head, and then there's this battery pack, and also a memory card provided by white matter, which is amazingly small. It can record for about 2 hours from these 128 channels. So I implanted 3 animals with these this device, and I've been recording from them. Oh, this slide is outdated actually. 1 of them for a year and 2 of them for about 4 months. Here's an example video of the animals. This one is wearing the logger. You can tell because it has these 2 little LED lights on the top. Otherwise, it's pretty small. You might not even notice it that much. And over here is just the kind of estimated depth signal from the depth camera. And so what you can kind of get a sense of is that the logger doesn't impede their normal behavior. You can see this guy is chasing around his mate. And he's still able to jump and play and do all the normal things that he would do. They still groom each other as normal. Once the other animal gets acclimated to it, it doesn't really even kind of notice it. Let's see, anything else notable here? Oh yeah, in this data set, the animals both have white colored hair. I instead just use the LED lights to distinguish them. The model can learn that just as well as blue hair color. Okay, and so the approach is to do these implants and then take data in the same way that I described before, with the camera above the cage to get this top-down view and label it. Now we extract data from whatever neurons are in contact with the silicon probe on that day. I generally get between 25 and maybe like 75 well-isolated neurons, sorted through kilosort and then manually curated with phi. And now what I started to look at is kind of how these neurons' activity is related to the very basic variables like whether the animal is moving, whether the animal is near the other, the animal's position in the cage, and also some of these more complex variables like the behavioral state and the behavioral transitions. So for instance, in this example recording of, I guess 100 neurons, you can see that there's a few that are really well tuned to the distance between the others, between the animals, a few that are really well tuned to movement. And these neurons tend to, they tend not to both be the same. So like these neurons aren't also these neurons. And that's an active kind of direction for me. So certainly I'll have more to say about that in the future. And with that, I'd like to thank my collaborators, Haran, Jitu, Rahir, Shivanji, and Olivia, my mentors, Goping and Bob, and my funding, especially from the Simons Center. I've been a Simons Fellow for almost 2 years now. It's about to end. And it's been really great to be in this program. And with that, I'll take any other questions from the audience.
Great. So any questions can still go through Q&A. Well, there's a
Will,
question.
you're on mute.
Oh, okay. So I have, how much can you learn about the neurons, and are the locations reproducible among marmosets? So regarding how much you can learn, I've been kind of studying the field and seeing what kind of best practices are. Basically, there's kind of two sets of practices. One is for people who have a lot of neurons, like thousands, and one for people who are kind of more in my regime of like a single silicon probe. The people with a lot of neurons kind of take like the principal components of the neural signal and relate it to like the principal components of the behavior. Like there's some really cool work off the top of my head from Karsten Stringer. I think there's probably Nature and Science papers about it. And then there's this other kind of approach where you take all of the behavioral features you can and then try and see which of them are informative for modeling the activity of like a single neuron or a single brain region, kind of like Anne Churchland's work. And I think I'm gonna kind of more follow in like the Anne Churchland mode of trying to take all of the behavioral variables and for each neuron try to understand which are contributing to its behavior. I have you considered adding any type of wearables to measure physiological activity from the animals, such as heart rate and sleep? So we do have cameras that have IR lights and we can measure their sleep, and the animals basically just sleep all night, like very reliably unless there's a pregnancy. And I haven't recorded the behavior or neural activity during the night, but that's certainly a avenue forward because we know that that's something disrupted in the ASD animals and even in the ASD macaques, as our collaborators have shown. With respect to heart rate, I haven't measured it on purpose, but there was one time where I got weird noise that was about like 200 to 300 spikes per minute of noise that I think might have corresponded to heart rate. But other than that, I have not recorded either of those. We have wearables on the macaques, and so maybe we'll convert them for the marmosets. Have you considered using 2 cameras to reconstruct a 3D trajectory of the marmosets? So currently I do have 2 cameras. They're just both mounted above the cage, and you can, or at least I'm estimating their 3D coordinates using the distance from those 2 cameras because they're kind of like your human eyes near each other, but not on top of each other. Any other questions? Can I comment on the genetic complexity, how difficult and standard it is to generate genetic ASD models consistently? I can't answer this. Maybe Gopin can answer this question. I don't know that much.
Yeah, so this is Gopin. So the technology is getting better and better. So the major restraint, restraint is not the technology anymore. It's the amount of animal available. So both the use CRISPR knockout or base editing efficiency are very high. And we have recently in the last year also, we have dramatically improved from the injected egg to implant to pregnancy. Now reach around 50% of implanted animal will be pregnant. So, but you still need a certain number of young female, pseudo recipient, and you need eggs. So as long as there are large facilities, enough animal, we have 250 or something, then you can do consistently making this knockout or knock-in model. So I think it will be, the field and the foundation and NIH are putting a lot of effort and resource into it to try to gear up so make these animals available to a lot of more people. And we are making our animals available also to others, to the research community.
We also have a question from Yang. Would your approach allow for profiling of behaviors from individual animals like stereopathies or postures? I think so, because I think that in the case of an animal who continually goes through the same sequence of behaviors, you would see that through the transition between states. And also, you might see that in usage of a certain posture more frequently in general. And so I think that that is possible. And I also have a question. What's the data variability across pairs? Would we need a big change to detect abnormal behavior from ASD animals compared to wild type animals? So there is some variability. I think that's why I've been emphasizing kind of drawing these histograms that describe the normal distribution of behaviors rather than focusing on like the average behavior. And then I think we'll have a lot of resolution in looking at how those histograms shift. Like, so for example, if we have a histogram for like how often the animal looks at the other animal, and it just like shifts over to the right, even if that shift is kind of small, I think if we just measure it very carefully, we'll be able to detect smaller changes. Okay. Thank you guys all for coming to this talk. And
yeah, I think it's, yeah,
we're
almost.
Thank you, Will.
Yeah, thank you, Will. It's a great talk. And thank everyone for coming to the talk.